In this video, we're going to talk about the stages of infection. An individual that's immunocompromised. This means that they're more susceptible to infectious disease. Extremely old age or extreme youth, like newborn, infant, would be immunocompromised. Any type of a genetic disorder that will affect the immune system, anybody who's recently undergone surgery or an organ transplant will have a weakened immune system. Pretty much anyone with any other type of primary disease like cancer, diabetes, could compromise the immune system. A true pathogen. This is a pathogen that can infect anybody, whether they're healthy or whether they're immunocompromised. So, for example, influenza virus could infect anybody. Malaria could infect anybody. So, true pathogen infects someone healthy or immunocompromised. Opportunistic pathogen, just like the name sounds, it'll infect somebody when it has the opportunity. So, this is a pathogen that will only infect someone who's immunocompromised or somebody who already has a primary illness. Some examples of opportunistic pathogens, you're going to hear a lot about Pseudomonas. Pseudomonas is a healthcare associated infection, and it's found in the hospital environment. In fact, a recent study showed that a lot of soap dispensers in the hospital contain Pseudomonas. Another one, Candida albicans. This is a yeast infection, and yeast infections typically infect somebody who's on a broad spectrum antibiotic that may kill off the normal flora and allow the yeast to take over. Again, another opportunistic pathogen. This is a really important term that you're going to see a lot for the rest of the semester. A virulence factor. A virulence factor is any trait that makes a pathogen or makes a microbe more pathogenic. And there are so many. So any of the things we've talked about already, like forming a biofilm, forming a biofilm that allows the pathogens to be sticky and to stick to the linings in the human body, capsules, which allow the microbe to hide out from phagocytes in the human body, pili, which allow the microbes to exchange genes for antibiotic resistance, any type of antibiotic resistance. Anything that will make the pathogen more dangerous to you is a virulence factor. Okay, so the first step in the infectious process is entry. Okay, so first the pathogen needs to enter into the human body. And there's two ways a pathogen could enter. Exogenous pathogens, meaning the pathogen's coming from outside the body. So this is what you would normally think about, like inhalation. You inhale respiratory droplets in the air, and they may contain like a flu virus. Insect or animal bite that allows the pathogen to penetrate the skin. Most pathogens need to get in through a mucous membrane. So eyes, when you rub your eyes in your hands, contain the pathogen, or through sexual transmission. So exogenous is coming from the outside. The second way a pathogen could cause disease, it could be an endogenous pathogen, meaning the pathogen comes from within. How this happens is when normal flora, either they get into the wrong part of the body. Another way endogenous infection can occur is if the normal flora goes out of balance. So for example, everyone has a little bit of yeast as part of their normal flora. And usually the bacteria outnumber the yeast and keep them under control. If someone's on a broad spectrum antibiotic, it could potentially wipe out all those normal flora, bacteria, and allow the yeast to overgrow, and then it'll become a yeast infection. So these are how endogenous infections occur. So like I said, the portals of entry include skin. Bacteria don't have the ability to just go through your skin because your skin is very thick, it's very dry, there's antimicrobial substances. But anytime there's an open wound or like a bug or animal bite, this is how bacteria can enter through the skin. Gastrointestinal tract, which is a very common portal of entry. Respiratory tract, another common portal of entry. urogenital tract, which mostly includes sexually transmitted diseases, and transplacental. This is passing from the mother to the fetus. And there are only select diseases that have the ability to cross that placental barrier. And those diseases we abbreviate as STORCH, which stands for syphilis, toxoplasmosis. The O is other diseases because they ran out of letters. So the other diseases are Hep B, AIDS, and chlamydia, rubella, cytomegalovirus, and the herpes simplex virus. So these are pretty much the only infectious diseases that can cross that placental barrier. The infectious dose. This is important. This is the minimum number of pathogens required to cause illness. And typically, the lower the infectious dose may mean the more easily the infection can spread. So some examples just to show you. Measles virus. It only takes one virus, inhalation of one virus, to cause a measles infection. So that's why measles is so easily spread. Another one, Shigella. Shigella causes a intestinal food poisoning similar to Salmonella. But Shigella, you only need to ingest between 1 and 10 cells, which is barely anything, to get the illness. Now, if you look down at the bottom here, Salmonella, on the other hand, it's ingestion of 10,000 cells to feel the illness. So this is a great difference in infectious dose. Once the pathogen has entered the host, it now needs to start the infectious process. It needs to attach, and then it's going to start growing, colonizing the host. So, in order to attach to the host, there are many virulence factors that a pathogen could produce. So, for example, bacteria usually will use Fimbriae or a glycocalyx, which is like a sticky, sugary coating to stick to the host. Now, protozoa will use cilia to attach. Bacteria do not have cilia. This is a protozoan attachment mechanism. Helminths, those parasitic worms, can use things like suckers, hooks, or barbs to attach. Remember, viruses will use spikes. So each type of microbe has their own attachment mechanism. It now needs to hide out from the host immune system. And there's a lot of ways that microbes will hide out from the immune system. Some microbes actually secrete enzymes to actually kill the immune cells. Some microbes use a capsule, okay, so they'll surround their cell with the capsule, and this makes it really difficult for the phagocytes to engulf the cell. Some bacteria, such as Mycobacterium tuberculosis, actually will grow inside of phagocytes. And so they'll actually survive in them and even grow in them. So that's another way they can hide out from being destroyed. Now that the pathogen has entered, attached, and they're kind of hiding from the immune system, the next thing that a lot of them are going to do is secrete toxins. It always depends on the pathogen, the type of toxin that they secrete. So a toxinosis is any sort of infection, harm due to a toxin. Some examples, a toxemia. Anytime you see emia at the end of the word, it's associated with blood. So a toxemia is when a toxin gets into the bloodstream. Another example is an intoxication. This is where someone ingests the toxin. So they're not necessarily ingesting the bacteria, or the bacteria may not even survive in the host, but they are ingesting the actual toxin, which can cause the symptoms of the disease. So, for example, botulism and many types of food poisoning are due to intoxication. There are two main classes of bacterial toxins. The first class we've already heard about before, endotoxin. And the second class is exotoxins. Remember from a few chapters ago, endotoxin is actually a lipopolysaccharide, and it's actually found in the cell wall of gram-negative bacteria only. And occasionally, some of the endo is, when you see endo, it sounds like it's in the cell, but it's actually in the cell wall of the gram-negative bacteria. And occasionally, when gram-negative bacteria die, it will shed. If it does get into the bloodstream, then this is what we call endotoxic shock, or sometimes also known as septic shock. And this is a life-threatening event. Because endotoxins are lipopolysaccharide, or LPS, they don't get destroyed by cooking, so they're heat resistant. In addition, we can't make any vaccines, so there's no preventative vaccine, and there's no antitoxin, meaning a quick treatment to the toxin. Now, exotoxins, they are not lipopolysaccharide, they are proteins. They are actually secreted by the cell. And many different types of bacteria can make these. So gram-negative may make exotoxins and gram-positive. Even fungus can secrete exotoxins. Now, exotoxins typically have a specific cell target. So for example, some bacteria secrete a neurotoxin, which will target the nervous system. Enterotoxins, which will target the intestines. One other exotoxin we've heard of already, a hemolysin. Remember, these specifically lyse red blood cells. So typically, exotoxins have a very specific target. Because exotoxins are proteins, they are heat sensitive. So typically, when you cook your food thoroughly, you'll kill off any exotoxins that are present. Also, because they're proteins, the immune system can remember these proteins very well. So we can make a preventative vaccine to help prevent exotoxin infection. A preventative vaccine against a toxin is known as a toxoid. So for example, the DTaP, which is diphtheria, tetanus, and pertussis vaccine. And then lastly, we can also make an antitoxin. So if somebody is infected with an exotoxin, we can quickly treat them to help prevent further infection. So for example, if someone stepped on a rusty nail and we suspected they could get tetanus, they would quickly get the tetanus antitoxin to prevent that infection by tetanus. So during an infection, there are 4 stages that the host is experiencing. And the first stage is the incubation period. This is from the time from when the pathogen first enters until the host is experiencing signs of an illness. So during this time, the host is not really, doesn't even know they're sick yet because the pathogen is preparing to start the infection. And the incubation period can vary depending on the pathogen. So for example, most like common colds or flu, the incubation period may be a few days. So it could be like 3 days. Or for something like a typhoid fever, could be 2 weeks even. Rabies, the incubation period could be a month or even longer. So it always varies depending on the pathogen. The next phase is the prodromal stage. During the prodromal stage here, this is when someone's feeling vague symptoms. So maybe, for example, you know, when you're kind of getting sick and maybe you feel a little tired, maybe a little back pain. So vague symptoms. Stage 3 is the period of invasion. So this is the main symptoms are occurring now. So if a person's feeling full-blown sick, they may have a fever. And this, again, could last varying amounts of time depending on the pathogen. Then the final stage is the convalescence period. And this is the period where the person is in recovery stage. So they're starting to feel better. Now, what's key about this is, depending on the illness, a person could be contagious during any one of these 4 stages. Somebody could still be a carrier of the disease even when they feel totally better and they think that that the pathogen's gone. They could still be a carrier here as well. So those are the stages of the infection. If the infection remains at one part of the body, so for example, like a boil that's localized on the skin, This is a localized infection. And typically, the microbe is going to cause infection at the site that it entered at. If the infection spreads throughout the body, it's a systemic infection. And typically, it's going to spread through the bloodstream, and it's going to reach multiple sites of the body. And a systemic infection is definitely very dangerous, especially if it gets in the bloodstream. Lastly, a focal infection is one that starts at one site and then it spreads to another. A classic example is maybe somebody has an oral infection in the mouth, and so it starts there, and perhaps they brush their teeth too hard and the infection enters the bloodstream, and that could go straight to the heart and cause an infection in the heart. So that would be a focal infection. If the infection is more than one type of bacteria, it's a mixed infection, or also known as a polymicrobial infection. So for example, maybe there could be a mixed infection in the intestines of more than one bacteria. A lot of times in the mouth, oral infections of the mouth involve more than one type of bacteria. A primary infection is the initial infection, which in most cases could be caused by a true pathogen. So for example, maybe somebody gets the flu or strep throat or something like that. So we'll say, for example, flu. Then a secondary infection is when a second pathogen takes advantage now that the person has an infection, primary infection, they're immunocompromised. A second pathogen takes advantage. So typically this would be an opportunistic pathogen. So for example, if somebody had the flu or strep throat and the respiratory tract was weakened, they could then get a secondary pneumonia infection. Another primary infection could be chickenpox, and by accident, especially in children, they could be scratching at the site of the chickenpox, and they could accidentally tear open part of the skin where they're scratching at, and then staph can enter through that opening in the skin and cause a secondary staph infection. So these are some examples of patterns of infection. A sign is objective evidence of a disease, meaning something that healthcare worker could observe. It's observable, such as a rash. A rash is observable. A fever, this could be measured with a thermometer. It's observable. Any kind of swelling. A symptom is subjective. So this is a feeling that can't be measured by a doctor, but the patient is feeling it. So for example, pain, weakness, chills. Many times when somebody has an infection, there's changes to the blood cells. Anytime you see leuco is associated with the white blood cells. So leukocytosis is an increase in white blood cells due to an infection. Leukopenia is a decrease in white blood cells. Another thing that could happen is septicemia. Septicemia is major, okay? This is major bacteria, a lot of them, that are replicating and growing in the bloodstream. So septicemia is typically serious. This could be a life-threatening event. On the other hand, bacteremia, this is a small number of bacteria in the bloodstream. So maybe the bacteria are not actively replicating in the bloodstream. It's just a small number, and it's not necessarily as life-threatening of an event. Same thing with a viremia. This is a small number of viruses that are in the bloodstream, but does not necessarily mean that they're replicating in the bloodstream. Occasionally, an infection is asymptomatic, or also known as subclinical, meaning the patient is experiencing no signs or symptoms. Just because one person is asymptomatic, they could still spread, so they still can be a carrier. And someone else that gets the infection from them might experience signs and symptoms. So this often depends on the host defenses, on the immune system of that person. Lastly, once the pathogen has gone through its full course, it's eventually going to exit the host. Typically, the portal of exit for that pathogen is the same portal of entry, the same way it got in. So for example, a respiratory illness that probably entered through inhalation will also exit through coughing or sneezing through respiratory droplets. An infection that infected the gastrointestinal tract is typically going to exit in feces. An infection that entered through the urogenital tract will probably exit the same way. So typically they exit the same way they came in.